Depression, sadness, and malaise appear in ancient medical texts since Hippocrates introduced the concept of melancholia. To this day, depression remains one of the most globally pervasive diseases. While more than 350 million people are diagnosed, depression is remarkably poorly understood. The symptoms are easy enough to imagine: sadness, low energy, loss of interest, and disturbed sleep, which must persist for a minimum of 2 weeks prior to a clinical diagnosis of depression. Although anyone can be diagnosed with depression at any point in life, the most commonly affected groups include those without health insurance, those with little formal education, and the unemployed. While men and women are commonly diagnosed with depression, women are twice as likely to suffer from depression. Treatment for depression varies by disease severity. For mild cases of depression, exercise and social support may be sufficient, while for moderate depression, psychotherapy and antidepressants are more common. In extreme cases, severe depression is treated with antidepressants and vagus nerve stimulation. Globally, antidepressant drug prescriptions have increased dramatically, with up to a 50% increase in 10 years in some countries, and sales generate approximately $12 billion per year. Many antidepressant drugs were designed based on the serotonin hypothesis, which suggests that insufficient serotonin, a neurotransmitter, is responsible for symptoms of depression. As we will see later in this presentation, recent conflicting research reveals significant gaps in this hypothesis. If we accept the serotonin hypothesis, we can begin to understand the function of the most commonly prescribed antidepressant drugs, selective serotonin reuptake inhibitors, commonly referred to as SSRIs. Let's begin with the molecule itself. Serotonin is a monoamine neurotransmitter that is produced in the brain from the amino acid tryptophan. In certain pathways in the brain, action potentials stimulate serotonin production in the terminal boutons of presynaptic neurons. Serotonin is then exported into the synaptic cleft and binds to receptors on postsynaptic neurons. The final step of the pathway is the most relevant to our discussion of SSRIs. After serotonin binds to its postsynaptic target, it is reabsorbed into the presynaptic neuron and recycled for future use. Most antidepressant drugs act on the last step of this pathway, reuptake.
SSRIs prevent reuptake of serotonin by blocking the interaction between SLC6A4, the transporter, and serotonin. Based on studies in bacterial models of the transporters, SSRIs likely bind between the extracellular gate and the EL4 hairpin of the transporter. Blocking this interaction increases the amount of available serotonin in the synaptic cleft, which can continue to bind to postsynaptic receptors and therefore generate stronger postsynaptic action potentials. The transporter, SLC6A4, is a sodium chloride-dependent integral membrane protein that is a member of the sodium neurotransmitter SymPorter family. Under normal conditions, as the transporter allows serotonin to pass into the presynaptic neuron, it also brings in a chlorine atom and releases a potassium atom into the extracellular space. When SSRIs are introduced, this process is inhibited. Although the precise binding mechanism between SSRIs and SLC6A4 is unclear, there are structural similarities between serotonin and some common SSRIs. As you can see, all of these molecules contain an amine group and an aromatic system. Furthermore, we see the conservation of a polar substituent, most often involving halogens, attached to a benzene ring. We also see the loss of the OH group that was present in serotonin. Due to these structural similarities, SSRIs are able to bind to the transporter that normally allows for serotonin reuptake. To begin our discussion of SSRIs, we should start with one of the earliest derivatives, imipramine. Imipramine was developed in the 1950s and was found to have a surprising antidepressant effect. It was patented as one of the first tricyclic antidepressants and is still used today to treat certain variants of depression. The discovery of imipramine's antidepressant effect resulted in research that eventually led to the development of modern SSRIs. A closer inspection of the possible pathways imipramine could have been acting on led researchers to determine that 1. Serotonin reuptake inhibition was a key element of the antidepressant effect, and two, that imipramine did more than just inhibit the reuptake of serotonin, but also inhibited many other essential neurotransmitters, including epinephrine, norepinephrine, dopamine, and acetylcholine. Furthermore, imipramine impacted normal functioning of histamine, BDNF, and sigma and mu receptors throughout the brain. However, as imipramine was one of the first compounds effective for treating depression, it was widely prescribed. As you might imagine, the lack of specificity led to a significant number of undesirable effects. The main side effects seen in patients taking imipramine are maladies to the central nervous system, ranging from headaches to dizziness and even seizures. The severity of these effects led drug companies to streamline their research and focus on one element of the pathway, serotonin reuptake. They discovered SLC6A4 and narrowed down leads that specifically interacted with this transporter as the binding target.
In the 1960s, Arvid Carlsson of Astra AB, a Swedish pharmaceutical company, and his research group began to develop a drug that bound effectively to SLC6A4. By focusing on this specific serotonin transport gene, Carlsson made use of target-based drug discovery. A number of promising kits were found, notably phennyramine and its chlorine and bromine derivatives. Carlsson decided to start with brompheniramine, and was then able to optimize it into zimelidine, which became the first SSRI. This was the first time target-based drug discovery was successful in discovering a psychoactive drug. Zimelidine was patented in Belgium in 1972 and finally released onto the markets of select countries, including Sweden, in 1982. Zimelidine travels through the gastrointestinal tract and eventually reaches the liver, where it undergoes oxidative demethylation and is metabolized into norzimelidine. Shortly after zimelidine came onto the market, a select number of patients developed Guillain-Barré syndrome, a disorder affecting the peripheral nervous system, causing paralysis that spreads throughout the body. Because of this, after only a year, zimelidine was removed from the markets. Not long after zimelidine was withdrawn, a replacement was developed named fluoxetine. Fluoxetine features a daily dosage about a tenth the size of zimelidine and also a significantly longer half-life. Fluoxetine taken orally is mostly absorbed by the gastrointestinal tract and has a bioavailability of 60 to 80%. Like zimelidine, fluoxetine undergoes oxidative demethylation, metabolizing into norfluoxetine in the liver, and then proceeds to be excreted in urine. The developers of fluoxetine, Eli Lilly, filed for a new drug application in 1983, and in 1987 the United States Federal Drug Administration approved fluoxetine under the brand name Prozac. Despite a scientific article naming fluoxetine as the first SSRI, it was in fact the second SSRI to become a drug, though it was the first to be sold in the United States. Fluoxetine has shown no indications of causing Guillain-Barré syndrome, like its predecessor, and has had great sales since its introduction to the market. By 1990, Prozac had become the most prescribed antidepressant and was earning over $1 billion a year. Fluoxetine, which is listed on the World Health Organization's list of essential medicines, is still in the market today and was the third most prescribed antidepressant in 2010, behind sertraline and citalopram, which are the next two drugs that will be discussed. The next major SSRI to come along was sertraline. Sertraline, developed by Pfizer, was approved in 1991 and was the second most prescribed drug in the United States in 2011, with approximately 37 million prescriptions. Sertraline is absorbed by the intestines and has a bioavailability of only 44%. It is metabolized mostly in the liver, where like zimelidine and fluoxetine, the amine group in the sertraline undergoes N-demethylation, forming desmethylsertraline, and is then excreted by the kidneys. Compared to fluoxetine, sertraline has a larger daily dose and has a comparable half-life in humans. Another major SSRI to hit the drug market in the 1990s was citalopram, patented by Lundbeck, a Danish pharmaceutical company. The United States Food and Drug Administration approved citalopram in 1998 with the brand name Celexa as a racemic mixture of the R and S enantiomers. However, researchers have since discovered that only the S enantiomer of citalopram is responsible for the antidepressant effects observed by the drug. Because of this, Lundbeck quickly developed and released escitalopram with the brand name Lexapro in 2002, which contained only the S enantiomer of citalopram. Both drugs are absorbed extensively in the gastrointestinal tract and have bioavailabilities around 80%. Moreover, citalopram and escitalopram are metabolized in the liver, where they undergo N-demethylation, losing a methyl group from the amine. Both drugs feature significantly smaller daily doses than preceding SSRIs, and have comparable half-lives to other SSRIs. Despite escitalopram containing a formula without the useless R enantiomer and a smaller dosage, escitalopram was prescribed fewer times in 2011, most likely due to the increased prices associated with the brand name drug. Lundbeck's patent on Lexapro expired in 2012, and generics have since been released. Here is a table containing the daily doses, half-lives, and log Ps of the SSRIs that have been mentioned in this presentation. As you can see, the daily doses of the drugs have trended downward through the years, with the exception of sertraline. Moreover, the half-lives have become significantly longer since the first SSRI, zimelidine, all with half-lives over a day. Log P values are pretty consistent across all of the SSRIs shown, except for sertraline, which violates Lipinski's rules, with a log P of 5.1.
As mentioned earlier in the presentation, some people are skeptical of the efficacy of SSRIs. This is partly because the perceived success of these drugs relies so heavily on the serotonin hypothesis. What the serotonin hypothesis states is that there is an X to Y relationship between lowered serotonin levels and predisposition to experiencing depression. Simply put, SSRIs is seen as the ideal solution to this problem. The idea is that there is a serotonin deficiency that SSRIs will reverse by increasing the abundance of serotonin in the synaptic cleft. As the field of neuroscience develops and we discover more about mechanisms within the brain, this ideal relationship begins to shatter. Conservatively, we can say this hypothesis does not fully encompass the complex relationship between depression and serotonin. Others have taken a more aggressive stance, some saying that there is no benefit to taking SSRIs at all. In one shocking study, a meta-analysis of antidepressants in the US revealed that there is an 80% duplicated placebo effect. This figure is staggering in comparison to drugs with more established chemical pathways. Taking this research into consideration, we know that there are problems with SSRIs, but realize that there remains a place for them on the drug market. The fact that they have been used for decades speaks volume to their perceived efficacy, and until more research is conducted, we cannot completely discount SSRIs. SSRIs are a drug class used to treat depression, and were developed based on the serotonin hypothesis. The serotonin hypothesis suggests that there is insufficient serotonin in the brains of depressed patients. Therefore, inhibiting serotonin reuptake through SSRIs should decrease symptoms of depression. Remember that SSRIs inhibit reuptake by binding to SLC6A4, the transporter that normally facilitates reuptake. Imipramine, an early antidepressant, cued researchers into a more in-depth investigation of the role of serotonin in depression and initiated extensive research in this area. Over the years, there have been many SSRIs released onto the market. Zimelidine was the first SSRI to be developed, but was quickly withdrawn due to serious side effects. Fluoxetine replaced zimelidine and featured a much longer half-life as well as smaller dose sizes, without the side effects. Sertraline was the next major SSRI to be released and requires a larger daily dose, as well as a significantly higher log P than other SSRIs. The last SSRIs we discussed were citalopram and escitalopram, the S enantiomer of citalopram, which had significantly decreased daily doses and are becoming some of the major SSRIs on the market. SSRIs are some of the most commonly prescribed drugs worldwide. The skepticism we discussed earlier in this presentation concerning SSRIs merits additional research, but the simple fact remains that millions of people rely on these drugs every day. Given the profound impact on quality of life they convey to some patients, perhaps a simple correlation is all we need to know for now.